welcome to the latest CSF podcast on axial spondyloarthritis. We'll be bringing you new episodes on a bi-monthly basis alongside our psoriatic arthritis podcasts. And we'll also be supplying you with monthly slide decks to help you keep up to date with the latest research and publications in the field of axial SPA. First of all, allow me to introduce myself. I'm Sofia Ramiro, consultant rheumatologist and senior researcher at Leiden University Medical Center and Zuiderland Medical Center in the Netherlands. With me today is Professor Xenofon Baraliakos, Professor of Internal Medicine and Rheumatology at the Ruhr University in Bochum and a senior consultant and scientific coordinator of the Rheumatology Center Rheumazentrum Ruhrgebiet in Herne, Germany. And we are also joined by Professor Atul Deodhar, Professor of Medicine and Medical Director of Rheumatology Clinics in the Division of Arthritis and Rheumatic Diseases at Oregon Health and Science University in Portland, United States of America. And of course, if you want to find out more about us and the papers we discussed today, please head over to the CSF website, www.cytokinesignaling.com. So Xenofon, over to you.
Thank you, Sophia. In our first paper, the authors evaluated the efficacy and safety of Tofacitinib in biologic bDMARD-naïve and TNF-IR patients with ankylosing spondylitis or radiographic axial spondyloarthritis. Our second paper represents the results of two parallel clinical trials that investigated the safety and efficacy of the IL-17A and F blocker Bimekizumab. Over to you, Atul.
Yeah, thanks, Xenofon, and thanks, Sophia. So the first paper is titled Tofacitinib Efficacy and Safety in Patients with Ankylosing Spondylitis by Prior Biologic Disease-Modifying Anti-Rheumatic Drug Use, a Post Hoc Analysis. So the background of this study is that in the US, at least, Tofacitinib is approved for the treatment of ankylosing spondylitis only after TNF inhibitors have failed to work, or actually have been tried before, I should say. So prior exposure to biologic bDMARD therapy in patients with AS may influence subsequent treatment responses and drug tolerance. So we wanted to look at a post-hoc analysis to look at the efficacy and safety of Tofacitinib by prior biologic bDMARD use in patients with active AS in the phase 3 study. So patients were stratified by prior biologic bDMARD use, and outcomes were assessed to week 16, where patients were randomized 1:1 on Tofacitinib 5 mg BID or placebo, and week 48, where the placebo group was switched, of course, to Tofacitinib at week 16. What we found was from week 4 to week 48, ASAS20, ASAS40, and ASAS5/6 response rates to Tofacitinib were greater in the biologic bDMARD-naïve than TNF inhibitor inadequate responder patients, as expected. This is numerically better in biologic bDMARD-naïve patients. But when you look at the magnitude, so if you look at the difference between the active drug and the placebo, it was pretty similar for biologic bDMARD-naïve and TNF inhibitor inadequate responder. The ASDAS responses and the least square mean change from baseline in the ASDAS and high sensitivity CRP was also numerically greater with Tofacitinib than placebo in both biologic bDMARD groups, naive as well as IR. BASDAI 50 response rates and LS mean change from baseline in BASDAI were greater in the biologic bDMARD-naïve than TNF inhibitor inadequate responder patient between weeks 16 and 48. And when we looked at the AS quality of life and SF36 physical component survey, the LS mean change from baseline was more prominent in the biologic bDMARD-naïve than the TNF inhibitor inadequate responder patient. The discontinuation rates of the study drug due to adverse event was numerically higher in TNF inhibitor inadequate responder subgroup, which is not unusual. Something similar has been observed with other drugs as well, other biologics, that patients who are TNF inhibitor inadequate responder somehow drop out sooner with for lack of efficacy or more adverse effects compared to those who are biologic DMARD naive. So in conclusion, Tofacitinib generally demonstrated greater efficacy than placebo for biologic DMARD naive and TNF inhibitor inadequate responder. The biologic DMARD naive patients had numerically higher responses, but the delta between the placebo and the active drug was pretty similar. And these response rates were sustained up to week 48. And the benefit-risk balance for Tofacitinib treatment was favorable in both biologic DMARD naive and TNF inhibitor inadequate responder patients with AS. Now, one thing which I must mention is the total study was 270 patients, and 77% of the patients were biologic DMARD naive. So the inadequate responder patient, it's a pretty small group, 31 patients who were biologic DMARD TNF inhibitor inadequate responder, 31 of them had received Tofacitinib and 31 were on placebo. So we are comparing small groups here, which is one of the issues here. But the importance of the study is this is the group of patients where in the US we will be using this drug because of this black box warning after the oral surveillance study. So what do you guys think?
Thank you very much, Atul, for this nice presentation of this important paper. I totally agree with you. And actually, you mentioned that in the US, we have the black box warning of FDA. In Europe, we have EMA warnings, but not all patients need to have been exposed to TNF inhibitors before being treated with JAK inhibitors. But current practice is that when a new drug class comes to the market, it tends to be more used in patients that have been previously exposed to previous drugs. So I think, therefore, it's very important to get this data in these specific populations and compare them within in this case, within the same trial, so the groups can really be compared. I think it's informative and important for our daily clinical practice. I think it's also what I would expect and what we see with several of other compounds, that the magnitude of the response is higher in patients who are TNF naive, TNF inhibitors naive, but that also happens with the placebo. And therefore, the deltas were similar in both groups, as you mentioned. So I think the data are aligned with our expectations. The comment I was going to make, I think you already alluded to it, I was going to ask you, do you think that have we investigated sufficiently this population of insufficient responders with this small number of patients, or would we rather have a specific trial in patients that have previously failed TNF inhibitors, as we see for some other compounds?
Yeah, absolutely right. I mean, this was very small numbers, 31 patients versus 31 patients. I mean, it's a pretty small number. I don't think, well, certainly I know that there is no study going on specifically in this population. To give you an example, Upadacitinib, a different TNF inhibitor, specifically did a study in TNF inhibitor inadequate responder patients. And that study showed pretty robust responses in TNF inhibitor inadequate responder patients. But that's Upadacitinib. Tofacitinib doesn't have a study. A couple of quick points I wanted to make was the risk of cardiovascular events in ankylosing spondylitis, radiographic and non-radiographic AxSpA patient, and also in psoriatic arthritis for that matter, because the study, there is no study like oral surveillance in spondyloarthritis, either in AxSpA or in PSA. And the question is whether can we use the same idea that or the same risks above age 65, those who are smokers, past history of cardiovascular disease, are these the people where we should be avoiding this drug, even Tofacitinib, even in patients with PSA and axial spondyloarthritis, even though there are no studies? And we recently actually have looked at this, and that paper is in production somewhere, not published yet, but there our conclusion is that looking at the risk factors in PSA patient and in axial SPA patient with metabolic syndrome and diabetes and hypertension, etc., we believe that we should use the same precaution that above 65 smokers, past history of cardiovascular disease, be careful. So
I agree with you. I think the same. I think the problem is the patients with cardiovascular risk factors, and we have seen post-hoc analysis from the oral surveillance and identifying the risk factors mostly associated that have higher risk, the ones you have mentioned, and therefore also patients that have had a previous ischemic event. Those have been identified as the most important risk factors. On the other hand, perhaps PSA is a little bit different, but if we think about actual SPA, it's more younger population, healthier with less comorbidities. So I think the risks are probably less in actual SPA, although we do not have an oral surveillance in our trial population, our patient population, as you mentioned. And I dare to unfortunately also add, we will not have most likely in the near future, because there does not seem to be the clear request for that from the authorities, and therefore it will not be conducted. So I think we will learn from practice, we will learn with time, and we will learn from RA as well, and where we learn how to identify more the patients that are at more risk. So we managed to stratify risk perhaps a bit better. What do you think about that?
Xenofon?
Yeah, well, I do believe that this is exactly the case. It goes along to the experience we've had with other drugs, with other issues. Just reminding all the time about, or myself also, about the fear of candidiasis when secukinumab came out. Yeah, and that nobody speaks about it anymore. So I think we will get. I would say we will get used to deal with it. And this, of course, is for the benefit of the patients, since they will automatically get that, I call it now a service, in terms of really being even in a safer place when we make choices that we would then be discussing with them. So fully agree. And therefore, I'm also not so much afraid, also not for the RA population, because even there might be the need to apply these drugs in those who may have some kind of, let's say, suspicious history of something. I think that's exactly what we've learned in the last 15, 20 years. And this will go on. And obviously, that's for the benefit of everyone.
Yes, totally agree.
Just a quick one more point, if I may. Last week I was in India attending the Indian Rheumatology Association annual meeting. And I've made this point before in this group that there are 10 or 11 tofacitinibs available. These are generic tofacitinib, yes. 10 or 11 brands. And this is by necessity. Rheumatologists in India use that because of the cheapness of this drug. Pill, easy to take. I mean, even storage is easy, etc., etc. And I was asking them about this. And of course, there is no good sort of data, sort of central data keeping or reporting of data to central authority for myocardial infarctions, etc. But people have seen exactly the same thing, that people who are older and smokers and etc., they have had bad problems and also infection risk. And I was urging them that they should really look at it much more systematically, because these data will probably come from places like India, where there is a large number of patients being treated with tofacitinib, and somebody needs to kind of look at this more carefully. So real life data is going to be important, as Sofia, as you were saying earlier.
Yes, totally agree with you. And I agree that in that sense, India is positioned in a unique situation of having a high experience with tofacitinib due to the generics, as you mentioned, and they prescribe it a lot. And to the numbers in the population as well, of course, they will have a high experience. So it would be good to indeed learn from them, and ideally from studies set up there. Actually, on a slight different note, but not on this topic, but I was not surprised, I was positively surprised to see at ACR that there was a trial, not a study presented by Indian colleagues on tapering Tofacitinib, and there are no studies at all on tapering JAK inhibitors yet. There was something that we identified in the recommendations as yet an unmet need, and therefore we did not recommend any tapering of JAK inhibitors, only of biologics. And in India, due to the high usage, they are indeed in a position to run this type of studies that I think will be very informative for the community. And it will take a much longer time to be run in Western countries. So it would be good if they can be in the lead of that. I agree. Shall we continue to the second paper?
Yep, Xenofon, I think.
Yes, thank you. It is indeed. We can go over to it. That second paper is being entitled Bimekizumab treatment in patients with active axial spondyloarthritis, a 52-week efficacy and safety from the randomized parallel phase 3 bMobile-1 and bMobile-2 studies. So they have two phase 3 studies, which are being called 1 and 2. So for the study background and for the preclinical studies that we've gotten, the knowledge we've got, we have seen that there is superiority of Bimekizumab in reduction of inflammation and pathological bone formation versus IL-17A alone. And the Bimekizumab has been approved by the European Commission for the treatment of non-radiographic AxSpA, and psoriatic arthritis. So in the studies that I'm referring to here, they're called again bMobile-1 and bMobile-2. These are two parallel phase 3 clinical trials that are evaluating the efficacy and safety of Bimekizumab. And this publication reports the clinical efficacy and safety of the compound in patients with active axial spondyloarthritis for up to 1 year, 52 weeks. In both trials, patients were randomized 1-on-1 to Bimekizumab 160 milligrams every 4 weeks or placebo, and the placebo group was switched to active treatment after the primary endpoint. This means at week 16, and the primary endpoint was ASAS40. Regarding the results, ASAS40 responses at week 16 were sustained in TNF inhibitor naive, and those patients who were pretreated with TNF blockers and were randomized with Bimekizumab at baseline. The rate of ASAS40 response at week 16 was significantly higher for the treatment group as compared to placebo, which in fact is something that is trivial and expected. Indeed, this was seen here. And similar results were also seen for the other outcomes, such as ASAS20 and ASAS-PASI responses over the same time periods. A substantial proportion of patients in both treatment arms also achieved BASDAI 50. And at week 16, there was a rapid reduction of high sensitivity CRP in the treatment arm as compared to placebo, whereas placebo patients showed similar effects when they were switched to Bimekizumab. And there were also reductions in morning stiffness, BASFI, AS-QOL, and also the other outcomes that were observed at week 52. So altogether, we can say that patients that switched from placebo to Bimekizumab at week 16 showed comparable improvements to patients given Bimekizumab at baseline already by week 52, that was. Efficacy was sustained and consistent in patients with non-radiographic AxSpA and radiographic AxSpA across the domains across both domains. This means of axial spondyloarthritis. And regarding safety, the safety profile was consistent with results from phase 2-3 studies in axial spondyloarthritis and phase 3 studies in psoriatic arthritis. And well, the question is, of course, what we gain from this information for our patients who are being treated in daily practice. On the side note, we do have in the meantime the approval of Bimekizumab. We are using it, I would say relatively largely for a new compound due to all this data. And I think also that we've seen so far that these results are just, roughly presented or briefly presented in some more or less bullet points are in fact also found in daily practice. So I'm also happy to see what you have observed by prescribing the drug, because it is important, similar to the discussion we've had before, to gain information from daily life.
Yeah, definitely. Totally agree with you. Thank you very much for this presentation. Responding to your question, I can tell you that in the Netherlands, we do not have yet approval for the rheumatological indications. It's ongoing, the process of request. We have some patients already from the dermatology because it's approved and reimbursed for psoriasis, but not on patients that we prescribe from our side, but the ones from psoriasis that are prescribed by the dermatology, I think the results are very positive also in daily clinical practice, as you are saying. So the ones that we get contact with. And I think with the new compound, what you just mentioned is very important, our experience in daily clinical practice. And in the meanwhile, as data is accumulating, that we have longer term efficacy demonstrated, the sustained response demonstrated. And I think that's mostly what this paper is confirming, that there is sustained response, which I think is very important. Making the bridge with the first paper, although I know it was not only the focus of your paper, but there was also the response analyzed in patients who are TNF inhibitor naive and insufficient responders. And as opposed to the data on Tofacitinib, we here see a similar response in patients in both groups, which is quite different and unusual compared to what we would expect. What would you think about this? Can we explain this?
Well, as always, in the last years, we can only explain things after having seen them. This means we cannot predict them. It's good and bad. It's good and bad because we are obviously, in the majority of the cases, positively surprised. So I think there is a different way of approaching the targets we have, both with Bimekizumab by targeting the F domain of IL-17 on top of or together with IL-17A, and with JAK inhibitors now, I would dare to say in general, because we have a much broader cytokine approach and inhibition. So obviously we may indeed be closing gaps here, gaps that we didn't have by single cytokine inhibition in the past with the biologic DMARDs we've had. And therefore that might be explained by the, well, at least anticipated mode of action. But again, the positive thing here is that this anticipation and that these ideas that have been there, they seem to be backed up by this data, that we do have both those who have not been treated with a biologic before responding well to the drug, but also those who have failed a single cytokine inhibition before be now also in a good shape, and obviously more patients responding to these new drugs simply because we're just covering a broader range of inhibition.
You know, I have a slightly different take on this, and I'm going to take a different disease, psoriatic arthritis, and a different drug, which is guselkumab. But we looked, so there were two trials in guselkumab in psoriatic arthritis in inadequate biologic TNF inhibitor inadequate responder patient. Their Discover-1 study was a combined study of TNF inhibitor inadequate responder and TNF inhibitor biologic naive patient. But they did a second study called Cosmos, which was purely on patients with psoriatic arthritis who had TNF inhibitor inadequate responder. And we actually had their baseline serum samples of these patients. So, and we compared that, and Discover-2 was a study of purely TNF inhibitor inadequate naive population. And what we found out was patients who were TNF inhibitor inadequate responder at baseline before they got guselkumab had much higher levels of IL-17A and F compared to psoriatic arthritis patients who were biologic DMARD naive, indicating, making a theory here, is that in some patients, the disease is driven. Of course, we know that IL-23 and IL-17 axis is very important, but in some people, that axis is more important than others. We are not there yet to decide which patient has which axis more important, TNF axis or 2317 but this is a very tantalizing result that patients had, those who actually were TNF inhibitor inadequate responder had baseline levels very high, and they responded pretty well to guselkumab. We are talking, I mean, Xenofon's paper, what we discussed was axial spondyloarthritis, different drug, etc. But getting back to the same idea that maybe IL-17 inhibitors are more effective in TNF inhibitor inadequate responder. But then Ixekizumab didn't really have a similar response. Ixekizumab in axial SPA in inadequate TNF inhibitor inadequate responder patients had less response compared to those. So this IL-17F may be important in these situations. And I'm curious to know, I mean, I thought that when EMA approves a drug, then it is available in Netherlands and Germany and etc. But clearly I'm wrong. It's not approved here in the US. So you said so, Sofia.
But it's approved by the EMA, it's approved in the European countries, but it's not reimbursed now automatically. So the countries need to go their internal process of reimbursement, and sometimes it's quicker, sometimes it takes longer. But there are countries that take even much longer. So, but that's the process. So officially it's approved, but without reimbursement, it's the same as not being available.
Yeah. So,
but regarding your comments, we don't for the rheumatological indications yet. But I would like to comment on what you were saying, because I was going to even to raise the provocative question of whether Bimekizumab is a really new mode of action or is an IL-17 inhibitor. And I think this point that we are discussing is already the first that can help us question whether it's the same mechanism of action or whether the dual inhibition isn't really a slightly different mechanism of action. And I think the other one is the signal on uveitis, as we see that with Bimekizumab patients seem to have less or a lower rate of uveitis, certainly compared to placebo in the first phases and then throughout the trial as well. And while with either IL-17 inhibitors, we do not see that. And we actually see that they are not efficacious, and especially data on secukinumab is available, not efficacious on uveitis at all. So this is also a distinction between molecules from the IL-17 inhibitors class. So I wonder whether this dual inhibition is not really classifying it as a different mechanism of action, and therefore being responsible for some slightly different effects that are reflected in this different efficacy. What is your take on that? Any of you can start.
Yeah, quickly. I mean, so theoretical what we think and then what practical you find is two different things. And yes, I mean, what I originally quoted that IL-17F blocking may be important in patients who are TNF inhibitor inadequate responder. Having said that, we recently completed a network meta-analysis of systematic literature review network meta-analysis, because there is no comparison between IL-17A inhibitors versus IL-17AF inhibitors, which is what the hidden question is this drug different than just an IL-17A inhibitor, Bimekizumab? So looking at when we did this network meta-analysis, we found, interestingly, there was no superiority of Bimekizumab, at least in the network meta-analysis, compared to
Atul, before just before, sorry to interrupt, but I'm doing this on purpose because of the type of the comment I will make, which is, to what extent can we believe network meta-analysis? So regardless of the results that you are telling. So I think that what we want is head-to-head studies. If the head-to-head studies are not there, are we now believing on artificial comparisons of patients that have not been compared? Different trial populations with differences between them. Some are completely TNF inhibitor naive, other are a mixed population. What are we comparing? Aren't we comparing apples and oranges?
No, you're right. And that's why in network meta-analysis, we specifically separated TNF inhibitor inadequate responder trials, mixed trials, and biologic bDMARD-naïve trials. And that's what I was going to say. We did that very carefully. And in the TNF inhibitor inadequate responder trials, there was no difference when you do network meta-analysis. And yes, there are limitations to network meta-analysis, but I doubt that any company will really do head-to-head studies between IL-17A versus IL-17A and F inhibitor, because
it was
they're
done in
expensive.
psoriasis, right?
Sorry?
It was done in psoriasis.
Yeah, they did it in psoriasis because they had surety that they probably will do better. If it is kind of close kind of stuff, then they're very unlikely to do in the. It's very difficult to show superiority in any for any drugs when it comes to musculoskeletal manifestations. So we actually looked at, when we separated out, we didn't really use we did not take the mixed population, and there was no difference, is what I was going to say. In fact, if at all there was a difference with secukinumab. Ixekizumab was not with ixekizumab. So bimekizumab was superior to secukinumab in the network meta-analysis in patients with axial spondyloarthritis who were mostly biologic DMARD-naïve, and that too in only a couple of outcome measures, ASAS-20 and ASAS-40, but not for high hurdle endpoints like ASDAS, inactive disease, and etc. So there are all these kind of stuff. When you say superior, non-superior, which outcome measures, what type of population, as you are saying, naive, mixed, inadequate responder, etc. So anyway, it's very exciting. And I mean, it's kind of, you make theories about this. And here again, our clinical experience will tell us whether this drug works very well in patients with who are inadequate responder to TNF inhibitor.
Any last comment from you, Xenofon?
Well, I see the comment from Atul. I understand that, yes, there might be different reasons for having inflammation or having issues with disease activity. And that is something that is extremely interesting as a theory, beyond or next to what we've just did. I made a comment or two in the beginning. I think both may exist or maybe even coexist. Yeah. It's really an interesting approach. We just need to see that by any kind of different studies that will come in the future. Unfortunately, indeed, I don't expect anyone to do a study with a direct comparison. This means head-to-head in the field of AxSpA, which is unfortunate, but we'll continue learning from the data.
Yes, definitely. I think we have had a very rich discussion, and we could continue for a long time, but I think this is bringing us to the end of this podcast. Thank you very much to both of you for this good discussion. And thank you all for joining us in this AxSpA podcast brought to you by the CSF. We really hope you find it useful. If you did, don't forget to subscribe to our channels on YouTube, SoundCloud, Spotify, or wherever you hear your podcasts from, so that you don't miss any future episodes. And if you want to read more about what we've discussed today, head over to cytokinesignaling.com, where you'll find summary slides of both of these papers. See you next time.